Welcome back, everyone. My name is Aleena Mushtaq. So we met yesterday, and I spoke to you about the overview of sort of like an introduction to the Ensembl genome browser. And then we looked at the location tab. So basically, we looked at genes in the context of their region along the genome. And then also we looked at the gene tab and the transcript tab, and I showed you how you can find different information on these tabs. Today, we're going to be talking about variation. And then I will talk to you about VEP, which is a tool that you can use to annotate your own variants. And then in the afternoon, later on in the afternoon, we're going to talk about comparative genomics and biomart. So just to recap, that all of the material for this training session, you can find that on training.ensembl.org. So this is our training site for Ensembl. And if you click on training materials on the blue navigation bar at the top, you can then find the specific page for this workshop. And on this page, you can find a PDF version to my presentation. So this contains everything from yesterday and also today. We also have a living document that, so this is a shared Google document that you can use to ask any questions, share screenshots if you're having issues with any of the exercises. And then we have these tabs for demos and exercises. So we've completed Ensembl species, genomic regions, and genes and transcripts. And today we're going to focus on variation, VEP, comparative genomics, and biomart. So again, the tabs, if you just click on them, they're going to have two further tabs, one for demo and then one for exercises. So the exercises, we have two exercises, for example, here, and the questions are followed by a text answer. So you can always work your way backwards if you'd like. Before I start, are there any questions from yesterday or any questions about logistics today? Okay, please do let me know if there are any questions. So let's start with the variation workshop. So we're now going to talk about the different types of variation data that there are and the different types of data that are associated with the variants in Ensembl. So we'll be focusing at how the different variants are classified. I'll talk about different species and the sources Ensembl draws variation data from. We'll explore the variation data through the Ensembl browser. So I'll do this in the demonstration after the presentation. And then we will also look at VEP, which is a tool that allows you to find information about your own variants. So variants can be thought about at different sizes or number of nucleotides affected at a time. So we can have SNPs, which are single nucleotide polymorphisms. We can have small deletions or insertions. Then there can also be large-scale structural variants, for example, deletions of 100 or so bases affected at a time. And we could also have translocation or copy number variation types. So variation is important for adaptation and evolution purposes. And some of the practical applications would be breeding of plants and farm animals, personalized medicine such as disease diagnostics and pharmacogenomics, and also forensic science. In Ensembl, we have about 23 vertebrate species with variation data. So humans are the most well studied, but we do also have variants identified across model organisms such as mouse, rats, agricultural species like cows and sheep, and obviously also Atlantic salmon. And there is also variation data available for some of the genomes in Ensembl fungi, metazoa, plants, and protists. So there are four main steps in Ensembl variation data pipeline. So the first thing that we do is that we import a number of resources, import from a number of resources, and then we apply quality control on this data that we import. And then we look at other databases to look for linked or association data. And finally, we analyze and process all of this data ourselves to make it available on the browser and other interfaces. So if we look at the first step, which is very important, we look for various publicly available archives and variation databases to import the variance data from. For humans, the most popular is dbSNP. For non-vertebrates, information would come from EVA. So this is European Variation Archive. DGV archive has lots of data for structural variants, and then Cosmic and Cancer's gene census for information about somatic variants. So here we have the source for Atlantic salmon. So you can see that we have the source just over here. And we have the description. So these are variants with genotypes imported from EVA. And then we also have some information on data types. So these are from VCF. You can find this information for various different species by going to the link on my slide. Then the second step is to apply quality control. And this is where we do things like mapping to the appropriate genome assembly. We check for alleles matched between reported and reference genome sequence. We perform check on the alleles of RefSNP and dbSNP submissions, and then we also check for external failure classifications. And the third step is linked data. So this is where we add value to our imported data. We look for link or association data for every single variant that we import in step 1. So we look at a number of different databases for additional information, such as allele frequency, associated phenotype and disease, a list of citations that might mention these variants specifically. So I have a list of few projects that we use for this type of data. And then the last step, sorry, I've just got a message for security update, but let's just ignore this. Okay, can you see my screen okay? If you could please let me know in the chat. I think it's okay, but it would be nice to have some confirmation. Okay, thank you, guys. Okay, so the last step is Ensembl analysis. So this is where we apply our own analysis. So Ensembl predicts variant consequences. So we look to see the effect of the variant upon the gene, protein function prediction, whether a variant will be deleterious for a particular protein, if it's affecting the coding sequence of a transcript, for example. And then we also calculate, this is for humans only, we also calculate linkage disequilibrium, and we also look at conservation across different species. So the way that the consequence pipeline works is that we predict consequences of the variant that we import into our database. So the process of how we predict the variant functional consequences is that we take each variant one by one and we plot where it falls along the genome. So if you imagine this black line as the reference genome, and where a variant falls in the context of a gene is then going to determine its functional consequence. So if you think of a variant, for example, that falls outside of a gene, so it might fall in this 5' upstream region or this 3' downstream region, in which case the variant might not directly affect the protein, but it might fall in the regulatory region, which will affect the transcription activity of that gene or its transcript. Then we can think of a variant that might fall within a gene, but not within the coding region. So it might fall in this 5' UTR region, the splice site, or intronic site. Also, in this case, the variants are not directly going to affect the protein structure and the function, but they might affect the mRNA stability, the splicing, the translation efficiency, and other factors that might lead to a change in the expression of the protein. Finally, we can think about variants that fall within the coding sequence of a gene, and this is depicted here in yellow. So these can come as two different types. Firstly, we could have a coding synonymous consequence. which is when a variant in the genome does not result in a change in the protein sequence. And we can get these because we know that the genetic code is degenerate. So we know that more than one codon can encode for the same amino acid. So what that means here is that there might be a change in the genome sequence, but it doesn't result in a change in the encoded amino acid at this position. On the other hand, we might have a change in the genome that does result in a change in the encoded amino acid at that position. And this is what we're going to call a missense variant or a non-synonymous variant. So for every variant, we predict where this variant falls along the genome, and then we're going to assign a consequence based on that position. The sequences that we assign to the variants have very specific terms. The consequences are all defined by sequence ontology. So just like yesterday, we spoke about gene ontology, which is standardized vocabulary of terms to define gene function. Here we have sequence ontology, which is a standardized vocabulary of terms to define sequence changes. So here as an example, you can see that this yellow color coding over here would indicate missense variant. And then we have a definition for this variant. So it's a sequence variant that changes one or more bases, resulting in a different amino acid sequence, but where the length is preserved. And then there's also a accession number for this sequence ontology. You don't need to memorize this table. Every page in Ensembl that mentions a variant is going to have a color coding next to it, and the colors are always going to have a legend. But you can find more information on the predicted data for variation on the link on my slide. The last thing I would like to mention here is reference alleles. So I mentioned yesterday briefly that a chromosome is made of contigs that are laid end to end, just like you see in this diagram over here. So these are different contigs. And a single contig is a sequence from one individual. So in this example, this chromosome is made up of 4 contigs, and each of these contigs comes from different individuals. So if we take a look at one of these contigs in detail, for example, this CM, we can see that this contig has a sequence, and we can identify the position that is highlighted in orange here, which is T. So the individual that was sequenced here as part of the genome project, where this contig is derived from, they're not actually very representative of the rest of the population around the world. So the T that this individual has is only represented in 5% of the population, whereas the rest of the population has a G at this position. However, T is the allele found in this sequenced contig. Therefore, T is going to be the reference allele, and G is going to be the alternative allele. And in Ensembl, we're going to display them as T/G. So the reference allele first / the alternative allele. So I just want to mention here that it doesn't matter about disease susceptibility or conservation. If T is the allele that is found in the reference genome, then that is going to be the reference allele mentioned. So it's not always necessary that the major allele is going to be the reference allele. I hope it makes sense, but please do ask, unmute yourself, ask questions if you have any. We're going to now move on to the demonstration. So we're going to look at the SMC3 gene in Atlantic salmon, and we're going to find the variants within this gene. And then we're also going to look at a region of SMC3 to find variants in that region. So I'm going to jump out of my presentation. And we're going to go to www.ensembl.org. And like we've done previously, we are going to select Atlantic salmon from this drop-down species. And I'm going to type in my gene of interest, which is SMC3. So let me also share this in the chat. And this is in Atlantic salmon. Once you're happy, let's click on Go. And here we have our search results. So I'm going to click on SMC3 Atlantic salmon gene. And we're on the gene tab. So we have explored this tab yesterday, but I would like to bring your focus to the genetic variation section on the left-hand side menu. So under genetic variation, we have data over here as variant table, variant image. If structural variants are available, then this will be, we will link you out to them. But however, this is grayed out, so it means that we don't have any structural variants data available for this. I'm going to click on variant table. So this is on the left-hand side menu. And this is now going to show me a table with all of the variants that fall within this MCC3 gene. So you can see that the table is quite huge. You do have the option to search this table. So you can search this table either by variant ID or alleles, a classification, transcript. You can also export this table by clicking on this Excel icon. And then you also have the option to filter this table. So you can filter this table by consequences, or you can filter this table by other columns of the table. So I'm going to filter this table by the missense consequence. So let's click on this consequence button. So I have all of the consequences over here, and I'm going to turn all off. And I'm going to click on missense variant consequence over here to turn only this consequence on. You can obviously select multiple if you'd like, but I'm only interested in missense variant that fall within this gene. Once I'm happy, I can click on apply. And now my table has filtered to show me the variants that have the missense consequence only. So in the first column, we have the variant ID, then we have the location, the alleles associated with this variant. So G is the reference allele and C is the alternative allele. We have the classification, so they're all SNPs. We have the source for them, so where these variants were imported from. And then we have some information on the consequence type. So we selected for missense variant. So that is the consequence that's given over here. And then we also have information on resulting amino acid. So in the first row, for example, we have a change of E to D at position 241. So this is the amino acid coordinate. And then in the last column, we have the transcript where this variant is, where this variant overlaps. To get rid of a filter, you can simply click on X that's next to the filter and go back to your original table. Now we're going to look at these variants in the location context. So as I mentioned before, that the location tab automatically opens next to the gene tab, so you can toggle between the two as we're going to do now. So I'm going to click on the location tab. And we've looked at this tab yesterday. So we have, this is basically a view of our region of interest. So our region of interest is shown in this red box. So we have 3 images here, and the next is in more zoomed in or sort of more resolution than the last. And then this third image is the most detailed image. So it's our entire red box. So you can see that by default on this image, we have some variants from this database. So this is PRJ EB34225. So we have some variants shown here already. But let's see if we can add some further data. So some further variation data if available. So I'm going to click on configure this page on the left-hand side. And I'm going to click on variation. And you'll notice that we only have one data track available for this, which is already turned on by default. So this is the Atlantic salmon EVA PRJ EB34225. So I'm just going to save and close this menu. And variants on this region, so these lines over here are depicted as variants, and you'll see that these lines have different colors. So all of these colors are basically coding for a specific consequence. We can find the legend just below the image. So these blue lines are intronic variants, whereas this peach or this salmon pink color over here that you see would be a splice region variant. The green over here is a synonymous variant, and so on. You can always click on a variant to find out more information about it. So I've clicked on this variant over here, and we have the ID for this variant, the classification for it, the location for this variant, the alleles associated, the ambiguity code, global minor allele frequency, consequence, and so on. If we click on this more about variant ID, so we have this variant ID over here. And if we click on this, this is going to take us to the variant tab. And this is what I'm going to do. Or alternatively, if you already know the ID of your variant, you can directly search for it in Ensembl as well. So we're now on the variant tab. And this tab gives us further information about this variant. So we have the variant ID over here. It's an SNP. We have the most severe consequence for it. So it's an intronic variant. And if you just hover over this title that says intron variant, it's going to give you description of this variant as well. So it's a transcript variant occurring within an intron. Then we have some alleles that are associated with this. So we have the C allele, which is reference, and then T, the alternative allele. We also have information on the minor allele frequency. We have information on the location where this variant falls, some HGVS names. So this is just another notation to describe your variants. So you can show all 7 variants by clicking on this plus icon. Sorry, all 7 HGVS names for this variant that are just shown here. We have information on the original source of this variant. So these variants are from the EVA study, and we have the study number over here, and we'll also link you out to it. And then we have information that this variant overlaps 6 transcripts, and it has 80 sample genotypes. You can further explore this variant by clicking on these different icons, or similarly, you can also explore this variant by this menu on the left-hand side. So it's basically the same information as the icons. To start with, I'm going to click on genes and regulation. And this takes me to a page with a table that shows me all of the genes and transcript consequences for this variant. So we have information over here on the genes that were affected. So it only affects one gene, as we know that all of these stable IDs are the same. Then we know that this variant overlaps 6 transcripts. We know this from this line where it says about this variant. So for each transcript, we have a consequence for it. So this is the first transcript over here, and we have a consequence with the T allele. And this is an intronic variant. You can see further details of this by clicking on show in this last column. And this is going to give you some more detail for this consequence. Okay, the next thing that I'm going to do is that I am going to click on population genetics on the left-hand side menu. And population genetics shows me allele frequencies across populations. So we have allele frequency for this variant over here. So this C is represented in 72% of the population, whereas T is represented in 28%. And then we can also view information about the subpopulation, which is this GASP for New Brunswick. We've got Penobscot River and St. John River as well. And then this information is shown as pie charts, but you can also find this information in a bit more detail in this table format. So basically we have this, we have all populations over here, and then we have the subpopulations shown below. And then the allele frequency is shown or represented as bar charts over here. We have information on allele frequency count, and then we also have information on genotype. So we have information on the percentage represented as C homozygous or CT heterozygous. Okay, so there is a lot more information available. So for example, you can find information for sample genotypes, and if phenotype data is available, we will show you the phenotype data as well on the left-hand side menu. We're now going to have some time for exercises. So if you go back to the training page, and we are going to be doing the exercises for this variation module. And we have two exercises over here. One of them is about exploring SNP in Atlantic salmon, and then the next one is on exploring sequence variant annotation in Atlantic salmon. So let's say that I'm going to give about 20 minutes for these exercises. And if there are any questions, please do let me know.